Good evening and welcome to the live practice session for the course Applied Environmental Microbiology. So this is the 10th week session, and in this session we'll be covering about solid waste microbiology and antimicrobial resistance. So before we start with today's session, I just want to confirm if there are any doubts from previous sessions. If there, you post in the chat box, or any other clarifications needed, you can post in the chat box. And also I'll share my screen now. So yes, I have shared the screen, so please confirm if the screen is visible and also the audio is clear. Is the screen visible and the audio clear? So I guess the screen is visible and the audio is clear. If there is any issue, please let me know so that I can reconnect. So the first question for today is the composition of generated waste depends on which of the following. So in this session we'll be seeing about solid waste microbiology and antimicrobial resistance. economy as well as a geography. So when we talk about this agricultural produce and the location of an industry and all, it depends on the geography. And demography also depends on geography, because if in extreme regions like a complete desert or a completely colder region, obviously your demographics is going to have a larger variation. And in other regions, valleys, plateaus, or temperate regions, tropical regions, there is going to be a distribution of population which can use a wide range of resources and generate different types of waste. So, demography, economy, and geography, all of the factors affect the composition of waste. Okay. Second one is composting is the only biological treatment for solid waste management. A very simple question. You just have to give me a true or false. You just have to give if the statement is true or false. Okay, this is a very simple question. Obviously, composting is not the only biological treatment for solid waste management. We have other aerobic and anaerobic biological processes, especially anaerobic digestion, widely used, and other treatments with microbes are also used. Okay, so the next question is, which of the following are not desired in a landfill liner? So now we are moving into landfills. Which of the following properties is undesirable in landfill liners? Any guesses? There are many options that could choose. If you have guessed one or two, you can just. Okay. So when we talk about a landfill, it is a engineered system. So we are creating landfills, so they are going to be in a particular uh, layered system, and we are going to have multiple layers, and to prevent uh, leachate production and methane gas production, this is not desirable in a landfill. So basically, when do we use landfill? When we have to dump the waste. We cannot do anything using the waste. So previously there are certain options, like either we go for some biological treatment for organics, or we go for some recycling kind of techniques for inorganics. If nothing can be done, then we will go for dumping the waste. So there are few ways that can be dumped basically. When we talk about the common solid waste that are getting landfilled, we talk about medicinal or biomedical waste. They come under hazardous waste, they have a separate landfill. Apart from that, from the normal day-to-day waste that we are using, we try to landfill this multi-layer plastics. So these plastics cannot be recycled, so we try to landfill them. So there are products that get landfilled, and similarly into the landfill there can also be the presence of organic waste, though we don't intend them, but there can be these organics in them. So typically in a landfill, the first or the bottommost layer that you would have is a landfill liner. So basically in the ground you are going to dig some kind of a big hole or something. Into that you are going to put your waste. So from this waste there can be many kinds of seepages into the ground and groundwater system. So to prevent that, because this is an engineered system, the first and the base layer that we have is a liner. So this liner uh, is like can be made of clay or bentonite or other geomembranes, geotextile fabrics and all. So there is a large number of research going on to find the most durable and strong liner uh, that prevents seepage of liquids or microbes entry into the nearby environment. And in that system itself you also have the leachate collection system, because if the leachate is being produced and not being taken out of the system, then that again is going to be um, more environmentally harmful. So in that case we also have a leachate collection system that is attached to the waste system. So in this case the landfill liner should be tear resistant, because you're going to dump a large amount of waste. The properties of the waste can also be very different. Like when we talk about this hazardous biomedical waste, there can be needles, syringes that can be thrown. So this should not damage the liner as well. So it should be tear resistant, both in terms of carrying the weight as well as the property of the object. It should be impact resistant. So this is more about the weight of the waste that we are loading, and functioning. So all these properties are desired in the landfill, whereas the landfill liner should not be highly permeable. So we are using a liner to prevent the seepage of the leachate into the ground and groundwater. also the solid waste from the incineration center is usually ash in most cases, and this ash again is only landfilled. Okay, very open-ended question. How will you classify waste based on treatment characteristics?
எவ்வளோ நேரம்
Any guesses? Exactly. So biodegradable and non-biodegradable. So which in other terms could be recyclable, organic, and recirculable. So mostly inorganic wastes or non-biodegradable wastes comes under the recyclable most of it. Like I told, the multi-layer plastics and other few other stuffs are not even recyclable, and they are not organic as well, and they are landfilled. So they may be termed as recalcitrant wastes, and the organic wastes are obviously biodegradable. And even in organic waste that are the organic waste that is very difficult to degrade again can be termed as recalcitrants. So recalcitrance is a property by which the substrate is very difficult to handle. So it can come in either of these classes. Okay, so the next question is, name a few of the landfilling methods that you know. Few types of landfilling methods that you know. So the answer is trench method, area method, commonly known methods. Apart from that, you have cell emplacement and canyon or depression method. So in the cell emplacement method, you this is this a cell is a basic block in a landfill, and waste gets deposited in one cell after the other, and they are compacted afterwards. Like it happens in every other landfill, the compaction is a volume reduction technique, and at the end of each day you cover it with soil. In trench method, um, you usually use it in a flat land. So here a trench is being um, constructed by excavation, and waste is placed inside the trench. Again, you create a trench by excavating soil, and the same soil you use to line in between the waste layers. And in area method, uh, wherever the trench method cannot work, So trench method is usually for flat land surfaces. In areas where the land is not flat, trench method cannot be used. Instead, you can use something like a area method. Here, the waste is spread and compacted on the ground surface itself. So here you don't have this soil material to line or cover the waste, and you must be procuring it from elsewhere. Uh, Apart from flat lands, in regions where the groundwater table is not so deep and the water level is so high, you use this area method. And also certain rocky regions where the underground rocks are more in number, you cannot go for the trench method, and instead you go for the area method. In canyon or depression method, the depression can be either natural or man-made, and you use such spaces to fill in the waste. These are some different types of landfilling methods that you use. Okay, so the next question is, in aerobic phase in the compost, which of the following are not produced? concentrated in anaerobic digestion process, so there is no generation of methane, and there is consumption of oxygen, but there is no production of oxygen, so that as well is right. And nitrogen is present in the compost, like in the form of nitrates or ammonium, etc., whereas there is no production of this nitrogen gas. This happens in denitrification process and all, but not in aerobic composting basically. Whereas you have this production of carbon dioxide and water. So the three products that are not formed during an aerobic phase of composting is methane, oxygen, and nitrogen. So only with denitrification process you have this nitrogen production. Okay. So this question, the pH reduction in the second phase of landfill stabilization is due to the production of volatile fatty acids. Okay. Which of the following is observed in the third phase of landfill stabilization? We saw that second phase there is going to be DFA production. So in the third phase of landfill stabilization, you can see increase in methane and increase in pH. So basically, second phase there was acid production, and in third phase there is going to be consumption of this acid, which will increase the pH because acidic consumption is there, and obviously methane is being produced in this phase, so there is increase in methane. There won't be increase in VHA because it's getting consumed, and there is no increased CO2 as well. So in the acidic phase of landfill stabilization, which of the following occurs? You can think logically and see which applies. acidic phase, typically the ammonium concentration increases. So first, when we talk about COD and BOD, both these parameters are not going to decrease, but it's going to increase. So we in acidic phase, the major product is going to be your volatile fatty acids. So in that case, the volatile fatty acids are becoming soluble forms of your COD, and there is not going to be any decrease in COD and BOD basically. They can be readily consumed by microorganisms to produce other products. Similarly, this is a fermentative process. So in the acidic phase, you have fermentation happening, which is why there is production of this volatile fatty acids and some amount of ethanol as well. So typically in a fermentative process, you have carbon dioxide production. So you have hydrogen and carbon dioxide production when it comes to this particular system. So the carbon dioxide concentration increases and not decreases. Whereas your proteins and other nitrogenous compounds in the waste uh, have this ammonia which is released into the system. So there is increase in the ammonia concentration, ammonium ion concentration, and that is the right answer for this question. I hope the explanation was clear. So because there is VFA production, your BOD and COD are not going to decrease. So that those two options are out of the range. Similarly, this is a fermentative process where carbon dioxide and hydrogen are produced, and ammonium is also produced. Ammonia is produced in this process from the nitrogen sources, and hence the increase in that concentration is the right answer. Okay, so the next question is about the leachate composition. Leachate composition depends on which of the following? waste is very fresh, then the organic content is going to be so high and active. In that case, you will have strong organic rich leachate, and based on the acidic pH of that, further leaching process also happens. So, but if the waste age is so high, then the leachate composition might vary because there will be obviously degradation of that organic matter, and the composition will vary. Sunlight and wind velocity don't play uh, any role in this process. pH could play a role, but it's not pH only, because when there is already leachate production, you will have multiple media phase that will reduce your pH uh, from what it was initially, and after that the leachate characteristics might vary depending on that particular process. So pH do affect, but it's not the pH only affect. Yes. So when we have this methanogenesis step, a sudden drop in methane production could be observed in the landscape. So if it is observed, what is the most likely reason why there is a sudden drop in methane production? Okay, if you know about this anaerobic process, you know that there is going to be bacteria and archaea. So until the acidogenesis process or acidification process, your bacteria is into play, which um, takes the fermentative route to produce all these volatile fatty acids. But when you talk about methane production, that could be another step or the last step in this process, where the archaeal species work in to produce methane by consuming carbon dioxide, hydrogen, and volatile fatty acids. So, in this case, when there is rapid fermentation, when the kinetics of the fermentative bacteria is so high, there is going to be a rapid accumulation of VFAs and a sudden drop in pH. Methanogens are so sensitive to this pH change and to very low pH that they are inhibited in the system. Either they die or all their metabolic processes is getting inhibited in the system. So therefore, if there is a sudden drop in methane production, we can assume that there is accumulation of VFAs. So this process should go in harmony. There should be hydrolysis and then fermentation should produce VFAs, and simultaneously this VFAs should be consumed by the archaea. If the rate of consumption of VFAs is slower than the rate of production of VFAs, then because of the sudden drop in pH, the methanogens are inhibited and there is going to be accumulation of VFAs. Because methanogens are inhibited, you can't see further stable or high methane production. So the next question is, in a landfill transitioning from acidogenic to methogenic phase, which change is most expected? So remember the explanation that I gave for the previous answer. Based on that, this question is just a rephrasing of the same answer. recirculation. Leachate recirculation improves landfill performance. That is a statement. Okay, that is the known. That if you are going to recirculate leachate, then the landfill performance is going to increase. What do you think should be the most probable reason by which leachate recirculation improves landfill performance? You can just take a guess based on your understanding. Any guesses? Okay, by recirculating the leachate, you are enhancing the moisture content and microbial content. So you have active microorganisms in the leachate. Once you recirculate, you are going to refeed them, and also the contact time and everything increases in that process. You also increase the moisture content because there is hydrolysis process which requires moisture, and enhancing moisture and microbial contact will improve the landfill performance. The other three options are not related. because it doesn't reduce microbial activity or it doesn't remove methane, and also it does not eliminate anaerobic zone as well. Out of the given four statements, identify which of the statements are false. Okay, so the statements are methane yield decreases in young landfills. No, because in young landfills you have this acidification and then followed by methanogenesis. So methane yield gradually increases but not decreases. Ammonia increases pH beyond optimal range. Not necessarily, it may or may not increase the pH beyond optimal range. And methanogens are bacterial species that generate methane. Methanogens are archaeal species and they generate methane, and leachate composition varies with waste age and other properties. So C and D are the false statements. the stabilization. So here you can see that first is the aerobic phase, uh, followed by the anaerobic phases. In anaerobic phases, you first have this acidic phase, then the methanogenesis phase, and again the stable methanogenesis phase. So if you can see, first let's talk about oxygen. So there is a steep shift from aerobic to anaerobic. So in the aerobic phase itself, your oxygen gets consumed and the composition like drastically drops down. And then when you talk about nitrogen, so there is nitrogen present and it gradually drops down uh, during the active methanogenesis phase, where the system is still unstable. The nitrogen concentration like nearly drops to nothing, and in stable phase you don't have any nitrogen. And then when you talk about hydrogen, hydrogen may not is not initially present in aerobic phase, but during this acidogenesis process hydrogen is produced, and again during methanogenesis process this hydrogen is consumed. So in that case, from the second phase onwards the hydrogen increases in the acidogenesis phase, and after that, because of the consumption by archaeal species, the concentration of hydrogen decreases. And then when you talk about I'm audible, right? Okay. Sorry for the connection issue. So as we were discussing, uh, we saw about oxygen, hydrogen, nitrogen, and we were seeing about methane. So methane production starts in the methanogenesis phase and gets stabilized uh, once the organic matter is consumed. So this is more similar to a batch process where when mostly organics are consumed, then there is very little methane production that's going to happen. And when we talk about carbon dioxide will initially increase and then decrease and then stabilize. So this is because during fermentation process carbon dioxide is produced, and after that methanogenesis can take either routes, which is hydrogenotropic or acetic-lactic. So in the process of hydrogenotropic methanogenesis, your CO2 combines with hydrogen, so that's why you see a decrease in hydrogen and carbon dioxide after their production in the acidic phase when they move on to the methanogenesis phase. But not all CO2 can get consumed. Your biogas is going to be a mixture of methane, carbon dioxide predominantly, apart from having ammonia and hydrogen sulfide as well. So it gets stabilized at some point, but it obviously should be less than the methane content. Similarly, if you see the graph in the bottom, uh, this has further classifications like aerobic phase, acidic acidogenesis phase, initial methanogenesis, stable methanogenesis, and if there is some air intrusion, how the different factors are going to happen, convert, and then methane oxidation phase, and then you can see the carbon dioxide generation phase and the soil air interaction phase. So these are the different phases that are present in the process. So you can see that the COD, BOD, all of it is going to be high until the acidogenesis phase, and then it's going to steeply reduce because there is going to be consumption of that by archaea to produce methane. It goes similarly with ammonia. You can see it's stabilizing at a point and then decreasing heavily during oxidation phases. So now the question is to differentiate aerobic, anaerobic and fermentative process. There is aerobic respiration, anaerobic respiration and fermentation. All these three are different. So how will you differentiate all these three processes? You can at least try to guess which factor could influence the differentiation. Okay, so the factor that differentiates is the electron acceptor basically. So in aerobic process, the terminal electron acceptor is oxygen, and we saw about this electron tower, right? So in that case, in aerobic process, you can see that the ATP production is going to be 32 ATPs. In glycolysis, you will have 2. In citric acid cycle, you will have 2, and in the respiratory chain, you will have 2. Because there is more ATP production, the net energy is going to be high, which is 979 kilojoule per mole of glucose. If the energy is high, then the microorganism will have more energy for housekeeping and for biomass production or cell replication. So that's why you can see a higher amount of biomass being generated from aerobic processes. So the sludge that is produced in aerobic processes like activated sludge process and all is very high because of this reason. On the contrast, when you talk about anaerobic respiration process, you have very less ATP production and henceforth the energy production. So in that case, the biomass generation is conserved here, and you don't see a lot of sludge being produced from anaerobic process. Instead, there is methane generation which leaves the system, and in between somewhere there is fermentative process where you have a medium amount of ATP generation, and the energy generation is also medium. So the key differentiating factor is the terminal electron acceptor and how that affects the ATP production. So because the ATP production is affected, what is the effect on the microbial growth and replication? So all this is highly interconnected, and when you design or choose some processes, you have to keep all of this in mind for designing the process. Okay, so aerobic degradation leads to which of the following? I guess this is a easy question. Okay, so the answer for this question is water release. In an anaerobic process, you have water consumption, methane formation, and sulfide formation, whereas in aerobic process, you have water release because the terminal electron acceptor is oxygen there. Okay, identify the true statements regarding the anaerobic process. Exactly. So in hydrolysis stage, aerobic bacteria is involved or fermentative bacteria is involved. So this depends on whether it is anaerobic digestion or landfills. And in the first statement, the wrong factor is that it's methanogenic archaea and not bacteria. Very simple. So now we move to the antimicrobial resistance part. We'll just see few questions from the very close for today. So match the following regarding AM.
எவ்வளோ நேரம்?
Okay. So right, efflex pumps pump antibiotics out, and beta lactamase helps with enzyme binding and resistant genes are found in plasmids as well, not only in chromosomes but in plasmids as well. When we do target modification, beta lactamase is for drug inactivation, and target modification uh, affects the enzyme binding. So all these are different mechanisms when we study about antimicrobial resistance. So this efflux pumps can pump out the antibiotics, and beta lactamase can bring about inactivation of antimicrobial drugs, which again indirectly constitutes to resistance conferring. And target modification, if you're going to modify the target, the enzyme mechanism is kind of a lock and key mechanism. So if there is a modification, then the binding and the enzymatic reaction will not occur. Okay, horizontal gene transfer in AMR is primarily facilitated by which of the following? I'll just walk through the remaining questions quickly. So conjugation is the horizontal gene transfer process where because we in the previous question we saw plasmids contain this resistant genes, and in conjugation plasmids are again transferred, and that is a horizontal gene transfer technique through which resistance genes can be transferred. Which mechanisms allows bacteria to survive multiple antibiotics? So as we saw, target modification can only allow bacteria to survive a specific antibiotic because you're going to specifically do some changes, and enzyme reactions are kind of uh, pair dependent. So it's not like you do the modification and multiple antibiotics don't work, not like that. Permeability also depends on the nature of antibiotic. Target modification and enzyme inhibition are correlated, so the remaining is the efflux pump. So efflux pump do not know which antibiotic you are using. When there are more pumps, more antibiotics are pumped out of the system, so that's very natural. So efflux pump is the mechanism by which bacteria can survive multiple antibiotics because they rapidly send out antibiotics irrespective of their type. Methane producing environments can influence AMR by which of the following? So by they promote horizontal gene transfer. Again, methanogenic environment is a multi microbe containing environment. There is high diversity of microbes, and therefore there is high chance of horizontal gene transfer happening so rapidly. So the last question is identify the true statements out of the following. So here landfill leachate contributes to AMR because it's high in organics, it has antimicrobial resistance genes as well as a diversity of microorganisms. And anaerobic conditions prevent gene transfer. So the other two statements are low antibiotic concentration have no effect on resistance, but they do have, and higher microbial diversity reduces resistance spread. No, it increases the resistance spread. So with that we will end the session for today. If you have any doubts, please post in the chat box, or thank you so much for joining.